Welcome to BioMindScape. Today we are going to know the production of prokaryotic messenger RNA. So the structure and expression of typical prokaryotic genetic coding for a protein. As we can see, the following event occur during the expression of this gene. With the help of a sigma factor, RNA polymerase recognize and bind to the promoter region. The bacterial promoter contain two consens sequence. They are known as the Tata box or the pre-mRNA box. So, and the 35 sequences. The promoter identify the starting site of the transcription and orient the enzyme on a template strand. The RNA polymerase separate the two strands of DNA as it reads the base sequence of the template strand. Transcription begins at the first base pair. Sigma factor is released as soon as the transcription is initiated. Then the core polymerase continue moving along the template strand in the 3' to 5' direction, synthesize the mRNA in the 5' to 3' direction. Then RNA polymerase eventually reaches a transcriptional termination signal, at which point it will stop transcription and release the complete mRNA molecule. And there are two kind of transcription terminators commonly found in prokaryotic gene. So row independent termination occurs when a newly formed RNA fold back on itself to form the GC rich hairpin loop, closely followed by the 6 and 8 uracil residues. So these two structural features of a newly synthesized RNA promote dissociation of RNA from the DNA template, and this type of termination we can see in the figure. So row dependent termination require participation of the row factor. This protein bind to the newly formed RNA and move toward the RNA polymerase that has paused at the termination site. Then row then displace RNA polymerase from the 3' end of the RNA. Transcription and translation can occur simultaneously in bacteria because there is no processing of prokaryotic mRNA, so no intron, and ribosome can begin translation the messenger even before transcription is complete. So ribosome bind to the sequence called the Shing dalgarno sequence in 5' untranslated region of the messenger of the message. So the protein synthesis began at AUG codon at the beginning of the coding region and continue until the ribosome reaches stop codon at the end of the coding region. So the ribosome translates the message in the 5' to 3' direction, synthesizing the protein from amino terminus to carboxyl terminus. So the mRNA produced by the gene follow as shown in the figure above is a monocistronic messenger, that is, it can transcribe from a single gene and code for only a single protein. The word cistron is another name of a gene. So some bacterial operon, for example lactose operon, produce polycistronic message, and in this case related gene groups together in a DNA are transcribed as one unit. So the mRNA in the case contain information for several genes and code for several different proteins. So the expression of prokaryotic protein coding gene involve the process which the information in DNA is used to produce a functional protein. So the gene expression in prokaryotic like E. coli consists of two main steps, the transcription and the translation. In transcription, it is making the mRNA from the DNA, and translation is making protein from the mRNA. So because prokaryotes do not have a nucleus, this process happens simultaneously, co-transcriptional translation. So in this figure, we can see the expression of monocistronic gene, which means a single gene is transcribed into one mRNA, which code for a one protein. So transcription, here there is a promoter region on the DNA containing a Tata box and other upstream sequences like 35 and 10 region, and this signal RNA polymerase where it begin the transcription. So RNA polymerase start transcription at the +1 site just downstream of the promoter. The coding region start at the ATG, that is starting codon, and ends at a stop codon, that is TGA. So the mRNA has 5' untranslated region that is the name here 5-UTR, which include the Shing dalgarno sequence which help the ribosome to bind, and a coding region which codes for a protein. So 3- untranslated region, that is 3-UTR, includes structure like stem lobes that help with the termination, and there is a termination which can occurs by the row dependent, that is GC rich hairpin followed by UUU sequence, and row dependent require row protein. So the translation the ribosome bind to the Shing dalgarno sequence in the 5-UTR. Now we know that what is a UTR. It is a 5- untranslated region. So this in translation the ribosome will bind to the SD sequence, and then translation began at AUG codon and ends up at the stop codon, that is UGA. So the ribosome read the message from 5- to 3- and synthesize the protein from N terminus to C terminus. Then there is another figure, that is a polycistonic gene expression. In this figure it shows the polycistonic mRNA, which is common in bacteria. One mRNA molecule contains several genes. 1 to 3 genes are present, gene 1, gene 2 and gene 3. Each have its own SD sequence and its own start, that is a Shing dalgarno sequence, and its own start AUG and stop codon. It have its own start and stop codon. The stop codon are various like UAA, UGA and UAG according to the gene respectively, gene 1, gene 2 and gene 3 respectively, and this allow the production of multiple different protein from a single mRNA. Example, lac operon in E. coli, and each gene is translated independently into a separate protein. So in this diagram what do we learn, that monocistronic mRNA, it is found in the eukaryote and some bacterial gene. One gene is equal to one protein. Then polycistonic mRNA, it is found in bacteria operon. One mRNA is equal to multiple protein. And Shing dalgarno sequence where the ribosome bind the site in prokaryotic mRNA which is absent in the eukaryotes, and transcription and translation are coupled in prokaryote. So this was all about the diagram of expression of prokaryotic protein coding genes. Thanks for watching. Do like, share and subscribe.